Good morning, everyone. I'm just talking about some exploratory, two exploratory project. And so I find this paper, this photo in my collections, and this is in 2008, before Bob officially joined Monell. So Bob and Li Kuan was a postdoc trainee in Bob's lab at Mount Sinai before he became a faculty member at Monell. And when I joined Monell, I studied and learned about chemical senses in Li Kuan's lab and Joe Brand's lab. So although technically I was not a trainee in Bob's lab, but I am from this scientific lineage from Bob Magolsky. So when I become a junior faculty member at Monell, Bob was my faculty mentor for many years. And I also learned quite a lot from Bob's knowledge of the field and his wisdoms, particularly in grant writing. So, and I went through my publication record, and I found I have 9 co-author papers with Bob on a variety of topics. So this is a demonstration of the collaborative environment at Monell. And the 2 of the project I'm going to talk about today also was done in collaboration with Bob Magolsky's lab. And we use the transgenic mouse strains generated by Bob's lab. And from the previous speakers, you know that many people in the chemical senses lab uses these transgenic resources generated by Bob's lab. And so I think that's a big contribution to the field. So Tom Finger already talked about the canonical TACE GPCR signaling pathway. I just go through it really quickly. So the pathway starts with the receptor, either T2R or T1R, and that when activated, signals through the G protein complex, PLC beta 2, IP3R3, that opens the N-channel trypan5, and that eventually opens the ATP channel COM1 and releases ATP, which is the neurotransmitter for TACE system. And so this canonical TACE signaling pathway is very well established in taste bud cells. And from previous talk, you also know that this pathway is at least partially conserved in extraoral chemical sensing cells. So the 2 projects I'm going to talk about will focusing on 3 components in this pathway, the T1R3, gasteusin, and trypan5. So the 2 topics, the first one I'll talk about inflammatory bowel disease, and the second one I'll talk about the T1R3 in a sepsis model. So inflammatory bowel disease, or IBD, is a chronic recurring inflammation of the GI tract. And it affects millions of people worldwide. And this is an outdated fake number. There are more people are being affected. And there are 2 major types, the ulcerative colitis that mainly affect the colon, and the Crohn's disease that could affect anywhere along the GI tract. And the treatment for these, for IBD, are anti-inflammatory drugs like corticosteroid, immune regulators, and biologics such as anti-TNF therapy. And but not all the patients respond well to this therapy, and some patients a significant percentage of patients will develop a resistance to the treatment. So there is still demand of new therapies. IBD is more common in developed country. So there is a hypothesis or theory that the reduced exposure to parasitic infection in developed regions may contribute to immune imbalance and increased risk of IBD. So I highlight the last point because this is really what motivated us to test signaling pathways in IBD. And this is a picture, illustration of top cells. You already hear quite a lot of it. So top cell is a type of epithelial cell in the intestine. This is specifically for the GI tract. So these epithelial cells share similarities of taste bud cells because they express taste signaling components like gasteusin and tryptophan. And these in the gut, top cell can detect parasites such as helminths and protozoa and produces signaling molecules. One of that is interleukin 25 or L25. And L25 acts on a type of innate immune cells, or LC2, that produces type 2 cytokine. One is shown here is L13. So L13 plays big roles in tissue repair and type 2 immunity that is really important for defense against parasites. And another similarity between taste bud cell, type 2 taste bud cells, and the top cells is their developmental pathways, because we know that PAL2F3 knockout mice lacks type 2 taste bud cells as well as top cells. So to test if the taste signaling pathways are involved in IBD, we use this chemical-induced colitis model. So this is dextrose sodium sulfate, or DSS. So if we gave the chemical in drinking solution to mice, after about a week or two, mice will develop a colon inflammation or colitis. And so this figure shown is the body weight measurement over time, and body weight loss is reliable symptoms that reflect the severity of IBD in this model. And you can see that the gasteusin knockout mice shows much more significant weight loss. And we also measure disease index, which is basically the index of two phenotypes. One is diarrhea, the other is intestinal bleeding. And again, gasteusin knockout mice shows much higher disease index. And after the experiment, mice was sacrificed, and then we measured the colon length of these mice. So IBD causes shrinkage of the colon, and you can see that in the top panels, the wild-type mice has longer colon, but gasteusin knockout mice have much shorter colon. And we also look at the spleen, which is secondary immune organ, and that the increased size of spleen is indication of more activation of the immune system. So gasteusin knockout mice also have much larger spleen. And so then we go on to look at the histology, to look at the tissue itself. And so you can see that these two panels are control mice that was not induced for IBD. And you can see that the structure of these mice are pretty much like wild type and knockout are pretty much indistinguishable. But after DSS-induced colitis, we look at different regions of the colon, and we can see that the bottom panels are from Gasthuson knockout mice. You can see the structures are severely disrupted. There are loss of the crypts in the colon, and you see epithelial eruptions in these mice. And although there's some changes in wild-type mice, but to a much less extent. So the other indicator of the inflammation are immune cell infiltration. So we look at these through immunostaining and using markers to different subsets of immune cells. And I won't go through the details, but by quantification, we know that CD45 is a general marker for white blood cells. And you can see there's significant increase in Gasthuson knockout mice for leukocytes, for neutrophil and macrophages. So again, showing these mice develop a more severe colitis. And the other thing we look at are the expression levels of different type of cytokines. So as shown here, the TNF and interferon gamma are typical, very strong inflammatory cytokines, and the so-called type 1 cytokines. And we know that patients with IBD has increased levels of TNF and interferon gamma. As I mentioned, anti-TNF therapy works, can be used to treat patients, but only subsets of patients respond to TNF therapy. And so, but in this model, we did see that Gasthuson knockout mice showed a much higher level of these two type 1 cytokines, TNF and interferon gamma. And the type 2 cytokine, the so-called type 2 cytokine that involved in type 2 immunity, including IL-5, IL-13, IL-10 is anti-inflammatory cytokine, these were all significantly decreased in knockout mice. So there is clear imbalance of type 1 and type 2 cytokine. And so next, we also tested tryptophan knockout mice in this model. I won't go through the details. The phenotype of tryptophan knockout mice are very similar, were very similar to Gasthuson knockout mice. And more immune cell infiltration, inflammation, as shown here. Just touch a little bit on this. As I mentioned, Pol2F3 is a transcription factor that is required for type 2 taste bud cell differentiation as well as top cell differentiation. And when we knock out the gene, the mice lacks the type 2 taste bud cells as well as the top cells. And so if our hypothesis is correct, that is the top cell is involved in regulating gut inflammation, we should see more severe colitis in these mice, and this is exactly what we see. So, all right. So I would not go through this figure in detail, but our results suggest that in addition to detecting parasites, so top cell may also involve in detecting tissue injury and in the gut. That's regulate tissue repair. And knockouts will cause more severe gut inflammation. And so just briefly touched on T1R3 in sepsis. The sepsis if is a life-threatening syndrome involve multi-organ dysfunction from dysregulated immune response. And at least 1.7 million US develop sepsis and 10 to 20% die from it. And patients who survive also develop a long-term physical and cognitive disability, and specific and effective treatment are still lacking. And so our study is really rooted from this publication that shows fasting is actually protective of bacterial sepsis, and supplement of glucose is detrimental. So because T1R3 is a nutrient sensor, so we want to test if T1R3 is involved. So we use this bacteria lipopolysaccharide model, which is simple and very reproducible. So we give wild-type mice two type of T1R3 knockout mice. One is generated by Bob's lab, the other generated by Ichiro's lab. And then we look at survival, immune response, and metabolic response. And so this figure shows clearly that when we knock out T1R3, the mice are protected in the LPS sepsis. And to confirm this, we use the T1R3 minus IM generated independently by Ichiro's lab, and we see the same protection. The next thing, we look at the different inflammatory cytokines. TNF is early release cytokine, and we can see it peaks at 2 hours in the blood, and we did not see a significant difference between the two mouse strains. And when we look at IL-6, it's much slower, has a much slower kinetics than TNF, and that we see at a later time point, we see a significant difference in IL-6 level. And we also did metabolomics. This is done at Pretty Core for analytical chemistry by Mike and Bruce. And we identified thousands of chemicals. Among those, 200 was significant. And these are the different clusters of the chemicals. No need to pay attention to details. I'll show the one of the cluster that has two of these B-type of vitamin. So you can see, especially in vitamin B5, that there's a significant difference in the post-LPS treated mice between wild-type and T1R3 knockout. So this is our model. So sepsis is caused by overwhelming infection, inflammation. It is well known that this infection inflammation causes metabolic deregulation, and often associated with insulin resistance, and drives glycolysis and breakdown of lipids and protein. And as a result, you will see this small molecule level goes up. And because TNF, these metabolic changes drives immune cell activation and excessive inflammation, which puts feedback to the metabolic pathways and contribute to multi-organ failure. And because T1R3 is a nutrient sensor, we hypothesize that it plays a role in this step and feedback to the metabolic dysregulation. And in T1R3 knockout, it will put a break to these two steps, and so that it's protective in this model. So finally, I just want to acknowledge the people who contributed to this and thank Bob for his long-time support of our research. And thank you. Sorry.
Thank you very much. Are there any questions? We have time for one question. Otherwise, we'll go to the other lunch. There's one question. Paul, it's a long way.
I'll just yell at lungs. work. I've been snorting. Let's see. So in humans, there's a general T1R3 inhibitor, which is lactosol. It doesn't work in mice, but could lactosol be a treatment in humans of sepsis?
I think that's a possibility, but we need to prove it in preclinical models. So recently I was talking to collaborators about possibility to, design small molecule inhibitor T1R3 in mice. But that's still in early stage, so we don't know the result.